Good morning. Namaste. This is Dr. Uma Kedvital, Faculty of Agricultural and Veterinary Sciences, Jyoti Vidyapeeth Women's University, Jaipur. I'll be taking the Agricultural Microbiology lab this semester. And as you know, in the previous sessions, we have covered the microscope, its principles, and all that. Just in the last session, we have been looking into the various methods for enumerating the microbial population, the bacteria, fungus, and actinomycetes in a soil sample. So we had stopped at the direct and indirect methods for counting of cells. So in the direct counting methods, we were using the various counting methods, the counting chambers, the hemocytometer and the Petrov-Hausen chambers, and also using the fluorescent stains like acridine orange and CTC, we were in order to measure the direct counts of microbes in the sample that we will be dealing with. And in the indirect measurements, there was a viable cell count using the plate, spread plate or the pour plate method, and also, excuse me, and also the most probable number method that will be using the gas production or the acid production by the strain that we'll be using. So then I had told you that there are other methods, that is the direct and the indirect measurement of microbial biomass. That is also considered to be methods for enumeration of microbial population. So in direct measurement of microbial biomass, we will actually be weighing the whole cells, like wet weight or dry weight of the bacteria, and that will be used. So we'll be basically centrifuging the cells from a particular volume and collecting it, drying it, or weighing it as such, and thereby getting an idea of how much biomass is present. And in indirect method, that will be like there are these biochemical components of each cells, like ATP is there, peptidoglycan that forms the cell wall of the bacteria, chlorophyll if we are looking into algae or plant cells, and there are also the turbidometric measurements that can be directly correlated to cell numbers. If we are having a standard curve, then we will be able to get the cell numbers. And then there are various methods, like this peptidoglycan is exclusive for bacterial cells, and then there are certain compounds that are like specific for fungal cells. Now, actinomycetes being in the archaea, they do not have peptidoglycan, but instead of the ester compound, they have the ether compound in them. So there are compounds which are like very specific for each class of the organisms, and we could use those specifically in order to determine the count of each of these organisms, each of these classes of organisms in our sample as well. So as the sample over here is soil. So as I was saying, if we are using a viable plate count, then we'll be using specific media. So one of the major disadvantages is that it is very specific, but that can be used to our advantage also. Like we don't want to know how much fungus is present. We only want to know how much bacteria is present, or we want to know only how much actinomycetes is present, or leave the bacteria and actinomycetes. We want to know only how much fungus is present. So depending on that, there are the different mediums. So for bacteria, it will be nutrient agar. The preparation you have already learned that. And then there are the cryptic soy agar that will have something called the cryptic soy broth medium, and agar will be added to it. Or there is the peptonized milk in which we will be adding some amount of cyclohexamide as well. And then there is a basic something known as a plate count agar that contains some amount of tryptone yeast extract glucose. and some salts. And that is also something like a nutrient agar, but it contains glucose as well. So as I had told you, when you're preparing medium for addition of glucose, make sure that you are not overheating it. And if there is a probability of overheating it, then make sure that the glucose is added only finally, because otherwise it will lead to caramelization and charring of the glucose due to Maillard reaction. Now, if we are using the medium for fungus, then there is something called the peptone glucose acid agar. So in that, what we will be adding is there will be glucose and peptone, definitely, but there will be the buffering salts as well. So KH2PO4, the potassium dihydrogen phosphate and magnesium sulfate, and all will be added. And then some amount of acid will also be added, because fungus grows better in the acidic environment, bacteria doesn't. Then there's a potato glucose agar. So in that we will be using potato. So what happens is fungus has got a lot more of degradation enzymes, and they will be able to grow on this. Like if you're keeping bread or potato outside, what grows is mold. You don't see actually bacteria growing on this, right? The first things that will grow on these food items is fungus, because fungus has the ability to degrade these polymers, the carbohydrates in the potato and all, and grow on it. So fungus can selectively grow on if you are using a potato glucose agar. And again, that will also have a pH regulation. Then there is another Martin's rose Bengal agar. So again, in that also glucose, peptone, and the salts will be there. And this also needs to be autoclaved. So now if we are using actinomycin, so that again, it will have starch and casein. Its requirements are slightly different. So you will be adding the nitrate salts like potassium nitrate and sodium chloride, and then again the buffering salts, the potassium monohydrogen phosphate and magnesium sulfate heptahydrate. So the growth of actinomycetes requires more complex mediums like calcium carbonate and ferrous sulfate. All these will need to be added, and the pH will be same as that of bacteria, but the presence of all these extra salts is more specific for the actinomycetes growth. So that is how these media will be selected for each of these purposes. Now, how to now if we are like, that will be the principle behind each of these, and how to select the media for what organism you have to grow. So if you want to do a viable cell count method, or if you are using a most probable number, then you will be using the broth, because the most probable number is not done on plates, it's done on broth in Durham tubes. So then you will be using these the broths. The agar component will be avoided, and the broths for these will be prepared. Everything else will remain the same. Now, if we are coming to the procedure part of this, so for the procedure, for the most the viable plate count, what you have to do is you have to take saline tubes and add like 9.9 ml of this one. For the first one, you will be taking 10 ml of the saline and adding your sample, 1 gram of soil to it. And once you have that, I'll share the screen. Yeah. So in this, you can see you have your sample. So this will contain 1 gram of soil. And once you have that, you will be adding like 0.1 ml to 9.9 ml. So that will be 1 in 100 dilution. So that makes for 10 power minus 2 dilution over here. Now, from the 10 power minus 2 dilution, if you are like mixing it well, vortex it or invert it like enough times, and then you are taking or pipette it very well so that it gets mixed completely. And from that, you are taking 0.1 ml and adding it to another 9.9 ml of saline. I'll give 10 power minus 4 dilution, because again, you are adding 0.1 ml to 9 ml, making it 10 ml. So 0.1 ml in 10 ml is like 100 microliters in 10,000 microliters. So if you are dividing it, there will be a factor of 100 left. 100 divided by 10,000 will give you 100. So the dilution factor is 100. That's why the 10 power minus 2 becomes 10 to the power minus 4. And then if you're taking 1 ml from here, so you will have the dilution of 10 to the power minus 4. Now, if you are taking 0.1 ml from here and adding it to 0.9 ml of saline and adding it to the plate of 10 to the power minus 5, then also, because you are diluting it, 0.1 ml plus 0.9 ml will give 1 ml. So you are adding essentially 0.1 ml, you are diluting it to 1 ml. So then that will give a dilution factor of 10 from the 10 power minus 4. So you have 10 power minus 5. You have the 10 power minus 6 cube prepared in the same way. So you get 10 power minus 6, 10 power minus 7, 10 power minus 8, 10 power minus 9. You will have all these plates. Because why are you not using 10 power, the original sample, or the 10 power minus 1 or 10 power minus 2 or even 10 power minus 3? Because you are using soil. And soil as such has got a huge microbial population. The expected count will be a minimum of 10 to the power 7 cells per gram of soil. So that will be the minimum what will be expected. So depending on that, you are going to get maybe if you are having 10 to the power 7 cells, then in 10 to the power minus 7 dilution only, you are going to get very few like cells over here. And in 10 power minus 6, you will be getting around 20 or 30 colonies. And in 10 power minus 7, you will be getting like less than 10 colonies. Now, if you have like 10 to the power 9 cells, here only you will be getting like very less colonies. Even over here, you will be getting 10 or so, or 10 or 20 or 30 colonies over here. And over here, you will be getting 300. That will be like too numerous to count. So what you are supposed to do is that you have to make these serial dilutions and then plate them. So each of them, you have to make sure that you are mixing it well so that the samples are evenly mixed and there will be an even number of cell population wherever you are like pipetting, and then otherwise you will be getting an erroneous plate count. And you will be using sterile pipettes, sterile Petri plates, and the entire work should be done in a laminar airflow chamber. And once this entire procedure has been done, allow for the plates to the liquid on it to dry in the laminar airflow. And so for that, you might have to keep it for some 10 minutes. And once that is dried, you have to close the lid, mark, and invert the tubes. And the tubes will be kept in the incubator in an inverted position. Why in an inverted position? What happens is, there will be some amount of humidity inside your incubator as well. So what happens is the water will get condensed on the top portion of the plate. And if you are keeping it in a normal position, this condensed water will fall onto your medium, and then it will either cause contamination or it will cause the bacterial cells to clump together, and you will not get an accurate plate count. So it is very essential that you keep the plates in an inverted position, and the normal incubation period in order to see a proper growth is about 48 hours, by which time, if there are cells in there, you should be able to get proper growth. And then there are the control plates. You have to keep plain medium and plain saline medium. So you will be putting one plain plate without any medium or any saline in order to check for the whether your medium is contaminated, and also one with the saline. So again, the same 1 ml of the saline alone without any sample being diluted in that will also be used, because if your saline itself is contaminated, and saline could be contaminated, then that will give you an erroneous count. So these controls, these negative controls have to be kept along with your sample. So that will be for the viable plate count. Now say you are for the result part, if you are getting 20 colonies in the 10 to the power minus 6 dilution plate. You got 20 colonies. Then what will be the number of cells in your original sample? So since the dilution is 10 to the power minus 6, your dilution factor is 10 to the power 6. So your dilution is 1 in 10 to the power 6. And so your dilution factor will be 10 to the power 6. And so you have 20 colonies in here. So you will have 20 into 10 to the power 6 or 2 into 10 to the power 7 cells. Rather 2 into the expression is actually 2 into 10 to the power 7 colony forming units per ml of the original sample or per gram, since you have weighed 1 gram of soil. So there will be 2 into 10 to the power 7 colony forming units per gram of your soil sample. And normally we are supposed to keep replicate so that and take an average of the number of colonies which are found on the plate. So less than 20 or 25 is not counted. More than 200 to 300 is also not counted. They will be kept as too numerous to count. Less than this number will give you an erroneous count, because if you are keeping triplicates and if you are having 20 colonies in one, there is always a probability that you will get only like 10 and 20 and 10, and you will be only getting like 5 in another. Then 20, 10 and 5, your error is going to be huge. So under such circumstances, normally a value of between 20 to 200 is preferred when you are having a colony, doing a viable plate count, and this count will be done on a colony counter, which was done. So now let us look at the most probable number. So most probable number, I had told you it will be done in a broth. So in this broth, I'll show you what we are doing. So we have the sample. Again, we'll be diluting it. So we'll have a 10 to the power. So we are having the 1 gram of soil in here. And then we will be having a 10 to the power minus 1, 10 to the power minus 2, and 10 to the power minus 3 dilutions. So this will have 9.9 ml of medium. Here, that is one of the major differences. Like we have the Durham tubes inverted in here. And so if there is any gas production that is happening, if we are looking for bacteria which have the ability to produce gas while they are doing the fermentation process, then that gas will be collected in here, and that can be visualized. So we will be able to find out the gas production in each of these, because this will be sealed in top. Once the experiment is set up, we'll be sealing it and keeping it. So we have the gas, if there is any gas production, or otherwise if we are instead of the Durham tubes, we could add a pH indicator and check for the change in pH. So those are all methods by which we will be doing the experiment. And once the experiment is done, we will be able to get the results of how much of bacteria or fungus or so the viable plate count is one of the best methods, because media are available for each and every one of these. So if you want to make for Actinomycin, you could use that complex medium that I had talked about earlier. The entire composition of the medium is there in the notes that has been provided to you. Or else there could be this nutrient agar if you are doing for bacterial culture, or the potato dextrose or the potato glucose agar if you want to isolate fungus. So that is the only major difference that will be there, because you will be using the selective media. Now, if you were to look for any of the pathogenic organisms that are involved in hemolysis, you could use a blood agar plate, or you could use McConkey agar plates if you want to do some selective isolation. So those are all methods that are available for selective isolation of bacteria, but that is for another the entire purpose is different over here. Like here you want to enumerate what is available. So the difference is that you can enumerate bacteria in one, you can enumerate fungus, you can enumerate actinomycin, but all together is very difficult because most of these viable plate count and such methods are very selective. So you could do them and then add them together in order to get a total estimate of how much total microorganisms will be present in your sample. Or if you are only interested in bacteria or only interested in the fungus or the actinomycetes part, depending on the requirements, you could always design your experiment. So your takeaway from this would be, in order to be able to identify how you will be selecting the media, what your requirements are, what you want to enumerate, how you want to enumerate it, what are using the available resources, and how you would go on with that. Your results will be your viable plate count, the number of cells that are available. Yeah, so I didn't tell you about the most probable number, how you will be doing the calculation for that, no? So there is a statistical table that is available. So for each of the probabilities, like for 10 power minus 1, if you have 0 cells and you have one, like you have kept 3 tubes, and then out of the 3, if you have only like 0 and 1 and 1 and 0, so what are the combinations that are available? Like if 10 power minus 1 gives no reaction, 10 power minus 2, in that one tube gives reaction, but in 10 power minus 3 the tube doesn't give reaction. So depending on that, we will get the most probable value number per ml and the statistical probability for that. So there will be a p-value of 0.1. This p-value, what it is, you will be learning in your biostatistics course if you haven't already. So depending on the p-value and the most probable value that is obtained from this table that will be provided to you, you are supposed to do your calculations and determine the most probable number. So that will be all for this session. Hope you have understood this. Go through the manual and read it properly, because this method is very essential even when you are doing a quality control for the food products or something like that. Also, you will be doing this viable count, because for the quality control purposes also you will be required to find out how many organisms are present. The enumeration of microorganisms is a highly important part of agricultural microbiology, and this will come in handy in the further future semesters as well. And if you are taking up a career in this also, it will be very essential. So you have to understand this is one of the most basic concepts and a very essential experiment. So kindly go through it very well and learn it very properly. That will be all for now. Thank you. Have a good day.